So first, thank you for having me, and sorry for the snafu this morning. Again, this is one of those pandemic days where nothing goes right. But I'm very excited to be here today and to be part of this wonderful group studying the minimal cell. So I'm without further ado, because as John mentioned, I have a hard stop because I have to attend CSR Council. I'll tell you a little bit about the initial steps of the work that we've done with these beautiful cells, and then hopefully I'll give you an idea of what can be done in the future. So I'll just start at the beginning, reminding ourselves that cryo-electron microscopy that everybody hears about all the time, that has structures being solved all the time. Typically, the one that people hear about is the one on the left, which is the single particle analysis, which consists of purifying molecules, combining many copies, hundreds of thousands of copies of the same type of molecule, and getting beautiful 3D structures, very often now at atomic level resolution. In the case of pleomorphic objects, such as an entire cell, of course, you can't combine 300,000 cells in 3D because at the molecular level, no two cells are alike. And in those cases, we use the modality that's called electron tomography, which is what we do a lot in my lab. And just to remind you very quickly what electron tomography is, it's exactly the same as a CAT scan in which we take pictures of the same cell in different orientations. And these images are combined into 3D, into beautiful images of cells at the molecular level. So just here to show you, this is a human cell. And what you're seeing is sort of like to show you that it's 3D, I'm going up and down. And the gray values basically correspond to the mass of all of the molecules. And we can do these annotations where we can look at filaments and membranes and a lot of very large molecules we can identify in a more or less automated way. So you can see here on the right, the nucleoplasm, and on the left, actin networks and microtubules, mitochondria, et cetera, et cetera. So in my lab, when I was a postdoc anyway, we established this workflow for cryo-electron tomography where we can start by freezing cells so fast that water doesn't have time to form crystals, which is why we can get these structures. And then we can combine this with fluorescence microscopy and then a technique called cryofib milling to try to make cells thicker, because basically, to be fair, the only type of cells that we can observe directly are cells that are about 300 nanometers. So basically, this 3A and a handful of others. And eventually we do this sort of tomography. So what's interesting to me in the lab is to really try to generate and test structural hypotheses in cellular context. You can use tomography as a structural tool, and we've done that in the past. But really the idea is to use it as a scaffolding technique to look at other results from other data, or experimental or computational data, and always tomography helps in this way. And so the thrust of my lab is really to try to move cryo-EM into the highest resolution imaging tool for cell biology, just as it is for molecules and structural biology. So my lab has a lot of different thrusts that all are centered about method development in electron tomography. So I'll tell you what we'll do here. Zan was kind enough to draw my attention to the, I mean, I knew of course about the syn cells, but to try to collaborate with John. And so Vincent is my student here on the left who would like exchange cells in the parking lot with John to get them frozen and imaged in the cell. And so basically we can do tomography of these cells. The JCVI-syn3A did not give us any of the challenges that we got with previous versions. And so here you can see two little cells. This is about a 200 nanometer scale bar, and you can see these cells look a lot like wild type or mycoplasma. And the idea in this first approach, as I said, was basically to try to provide information for, you heard probably Zane earlier today or last week, I can't remember, showing how this was going to be used to like figure out where the chromosome was originally. And what we can typically do for this is to find out all the ribosomes. This is a sort of quote unquote very easy task, and also the membrane. So to give the simulations boundary conditions, this was sort of like our humble contribution to that. And I just want to point out that typically, and most of you probably have seen this beautiful work by my very good friend, Julia Mohammed, in which you just take 1,000 tomograms of cells and then grabbed all the ribosomes and got these beautiful high resolution structures. Even could see the antibiotic bound to the ribosome, which was just fantastic and breakthrough work. What we were trying to do here was a little different, because normally when you want to do structure, you basically pick all the ribosomes you can. You don't really care when they come from or you know, if they're good or bad. You just basically drop them along the way. If they're not perfect, you drop them along the way and then just combine many different cells. And so that doesn't necessarily give you all of the true ribosomes, just the best particles to get high resolution. And what we wanted to do, of course, was to get the true positive ribosomes within a particular cell. That is to say, where everything else was excluded. And as simple as it sounds, it's actually not necessarily a simple task. All right, so the way that Vincent did that is that originally he picked 200 ribosomes manually. As you can obviously see from this picture, all of these big dots are ribosomes. So you can pick them and then align them and average them, and what comes out is a ribosome. So that's easy enough. So this is a very low resolution ribosome that he got, and he used this as a template. And the reason to not just use a PDB as a template is that you can bias what you pick, and the data can be a little different. So we found over the years, me and others, that this is the best way to do it. So, and as you can see, it's sort of like very few particles. Once he had this template, he went in and sort of very roughly tried to find all of the ribosomes within one single cell. And then you obviously end up over picking ribosomes. You pick things that are large and round and dark, and some of them are not going to be ribosomes. And then you get a ribosome structure from all of the tomograms that you have, and that you can get like a slightly better structure of a ribosome. And then you start to do classification in your tomogram. And basically, he figured out a clever trick, so to say, that he did classification, and as long as the ribosomes were in a good category, sort of in a large class, they will be deemed particles that are ribosomes, and then the rest he would just toss and then integrate that until he basically decided that all of the particles that they had were ribosomes. And I think Zane told you about once that happened and we had all of the ribosomes in place that we were sure that were true positive ribosomes, that was used basically to do the chromosome calculations. So obviously that's just the beginning, and there's a lot more that can be gauged from cryo-electron tomography. Just right off the bat, in a relatively automated way, you can get large biomolecules, ribosomes, as I just explained, but also polymerases. Hopefully over here in the bottom left, I don't know if you can see my cursor, you can see like a little filament that's sort of like trailing the membrane. These are sort of like very sort of straightforward and visible things to do. You can, in the context of this, also use CLEM. We're excited about working with TJ, who's completely devoted, or so he tells me, to get super resolution, even within these cells in cryo conditions that will be able to correlate directly. And we are in the process of developing some tags native to tomography in my lab. For the syn cells, we're not entirely sure how we would be able to put them inside, but at least, there's a lot of interesting things, as people have noted out, in the capsule and membrane proteins and things like that, that we would definitely be able to locate with tagging methods for tomography.